Hello, everyone. I'm LinXing Chen from the University of Science and Technology of China. Today, I'm going to introduce a prevalent jumbo phage clade that we found in human and many animals' gut microbiomes. So first, a quick background. As you may read from many virus-related papers, viruses are the most abundant biological entities on Earth, and they play many important roles in different ecosystems. As you can see from the tree of life in the middle, viruses can infect and kill their host, and they also can be divided into those infect bacterial viruses, also known as bacterial phages or phages for short, and also those for eukaryotic viruses and also archaeoviruses. So by function, they can infect and kill their host, and more importantly, they can also change host metabolism through AMGs and also help gene transfer between organisms. Most phages have very relatively small genomes, around 50 kilobase pair that encode 40 to 50 genes. But there is another group we call jumbo phages, which are much larger, often over 200 kilobase pair, and encode hundreds of genes. I have to mention that this definition is mainly based on genome size, so it is somewhat arbitrary rather than based on a clear biological feature. Many of these genes that encoded by jumbo phages are still unknown, so we know much less about how this phage works. The other thing, another limitation is that because jumbo phages are very hard to study, because they are so large, they cannot move well in soft agar, so they often do not form visible plaques. Without plaque formation, it becomes very, very difficult to isolate them using traditional methods. At the same time, metagenomics approaches also have limitations. As you can see from the right, virus sequences are highly diverse, and many regions are very different from non-reference. So it is often difficult to assemble long and complete viral genomes. And this is especially true for jumbo phage because by definition, they have very large genomes. So incomplete assembly can easily miss important parts of their genome. So overall, many jumbo phages remain poorly characterized. Actually, the study of jumbo phages has started since long ago, but before 2020, there were very few jumbo phage genomes that are available. And in this review published in 2017, as you can see the tree on the left, they summarized all the available jumbo phage genomes at that time, and there are only a total of 94 isolates and a few from metagenomics. On the right, I show a comparison between a typical small phage named lambda phage and a jumbo phage, phage G, that has a very large size, as you can see. So the jumbo phages is much large, both in genome size and in particle size as well. More recently, it's also in 2020, metagenomic study have greatly expanded our view of jumbo phage. For example, in this study led by Jill Banfield, we identified hundreds of jumbo phage from a wide range of environments. In total, we recovered about 350 jumbo phage genomes from thousands of metagenomics. And some of them are extremely large, with genome size exceeding 700 kilobase pair. So for comparison, the largest isolated jumbo phage is shorter than 500 kilobase pair. Importantly, these phages are found across many different habitats, as I just mentioned, including soil, water, and host-associated environments. So this study showed that jumbo phages are not rare, but actually widespread in nature. To better understand jumbo phages, I I forgot to mention that in the study, we also give them another name. So jumbo phage equals to huge phages. So to better understand huge phages, we constructed a large reference data sets, which we call the huge phage genome collection, or HPGC in short. We collected genomes from public datasets and previous studies as well. And after filtering, very careful filtering and removing redundancy, we obtained over 7,000 non-redundant huge phage genomes. These genomes were then grouped into species-level cluster. Importantly, these data sets include samples from different environments and is mainly composed of those from animal digestive systems and freshwater. So over these data sets allow us to study huge phages or jumbo phages at a much larger scale than before. Using these data sets, we next asked whether there are dominant huge phage groups in the gut. We first clustered the genome and 95% identity across the genome, and then focused on the larger clusters with more than 10 non-redundant genomes. Among these large clusters, as you can see from the bar plots, two were especially interesting because they were clearly enriched in gut samples, and more than half of the genome in these clusters came from the human gut. When we further group relative clusters together using a lower similarity cutoff that is 90%, we obtained 104 genomes in total into the same cluster. This suggests that we are not looking at just a few independent genomes, but a relatively large gut-associated jumbo phage group. Thus, we named this group JAG phage, just for jumbo gut phages. To further explore the diversity of JAG phages, we expanded our search using PepScot, a very useful tool. Starting from the initial 104 genomes, we were able to identify additional related sequences, increasing the total number to 139 genomes. We then examined their relationships and found that these genomes can be divided into several groups, which we refer to as alpha, beta, gamma, and delta. Interestingly, some of these groups, such as alpha, gamma, and delta, showed very high overall genome similarity across most of the genome lengths, but relatively no similarity in key viral mark genes like the large terminase. In contrast, other groups such as alpha and beta, they showed much lower overall genome similarity, but high large terminase identity. So overall here, these results suggest that JAG phages are more diverse than we initially thought, but still form a coherent and distinct group. To further examine how widespread JAG phages are, we use the large-scale assembly from the Logan datasets, yet another very useful metagenomic analysis tool. From this data, we were able to identify more than 1,500 JAG phage genomes. When we mapped the distribution, we found that they were actually present across many regions around the world. Notably, this sequence mainly come from human-associated samples, especially gut microbiomes, and also from related samples such as wastewater treatment. In addition, this analysis allowed us to identify many individual studies with a large number of JAG phage positive samples. This provides a valuable resource for more detailed and targeted analysis in the future. Next, we asked how abundant JAG phages are in the gut microbiome. So we mapping the metagenomic reads. We found that JAG phages account for about 1.1% of total gut metagenomic reads on average. Although this number may seem a little bit small, but it is actually quite substantial given that the high diversity of the gut virome. In some samples, JAG phage can reach much higher abundance and can even be more dominant than the well-known groups such as the cross phage. In addition, based on CRISPR-based matrix and co-occurrence patterns, we found that JAK phages likely infect bacteria from the general Bacteroides and Phocaeicola. These are two well-known genera that are typically abundant in the human gut. So overall, these results suggest that JAK phages are not only widespread, but also ecologically important in the gut microbiome. Next, we explore whether JAK phages can be transmitted between individuals. To address this, we analyze data from fecal microbiome transplantation, or FMT. We found that identical JAK phages present in the donor can also be detected in the recipients after transplantation. Importantly, these phages are not just transient, but can persist in the recipient's gut for a relatively long time, up to 14 to 25 weeks. So this suggests that JAK phages are not just present in the gut, but can also be stably maintained and transferred between hosts. Then we tried to reveal whether JAK phages are active in the human gut or not. We used metagenomic data. We found, metatranscription data. We found that JAK phages are transcriptionally active across multiple individuals. In fact, in some samples, we found that the transcripts from JAK phages account for a large fraction of total viral transcripts and can even exceed those from all other coexisting viruses in the human gut. This suggests that JAK phages are not only present, but are highly active in the gut environment. In addition, when we examined individual genes, we found evidence of co-transcription and RNA processing, even for genes that are split into several pieces in the genome. This indicates that these phages have very complex transcription regulation and gene expression mechanisms. Finally, we look at what interesting function JAK phage may encode. We then identify a calcium-translocating P-type ATPase gene in these phages. While protein structure prediction, we found that structurally, this protein is highly similar to long ATPases from bacteria and even humans, as you can see from the above, suggesting that these genes encoded by JAK phages are likely functional. Importantly, this gene is also transcriptional active, and in some cases, its expression level is even higher than that of their bacterial homologs. So this suggests that JAK phage may actively influence calcium transporter in their host. Overall, this provides a potential mechanism by which these JAK phages could modulate host physiology, but so far we're not sure about the exact consequences. So in summary, this study identified JAK phages as a dominant but previously overlooked components of the gut virome. They're huge phages with genome around 360 to 400 kilobase pair and are widely distributed across human and animal gut microbiome. They are also transmissible with evidence from mother to infant pairs that I didn't mention here, and fecal microbiome transplantation suggesting that they can be frequently spread across different hosts. Functionally, JAK phages are highly active in situ and can dominate the viral transcript pool, yet they show limited expression of lytic genes. I did not detail here, but you can find the information in our paper. So this indicate that they are not primarily in a lytic state. Notably, they encode a calcium-translocating P-type ATPase that is highly expressed, suggests direct manipulation of their host physiology. Overall, this work suggests that huge phages can act as active modulators of gut microbiome ecology. So Let's go. Thank you for your attention.